Hello everyone. I would first of all like to thank RedChip for organising today's presentation. My name is Dr. Jerry Canellis and I'm Chief Executive Officer of Immuron Ltd. I'd like to spend a little bit of time today to walk you through some of our company key highlights that may be of interest to you. First of all, please take time to review our forward-looking statement, which you'll find on slide 2. Slide 3 is a snapshot of where we are as a company. Immuron itself is a clinical stage biopharmaceutical company targeting inflammatory mediated and infectious diseases with an oral immunoglobulin platform technology. The technology itself is validated. We have one registered asset generating revenue and have 2 lead clinical assets in phase 2 development for the treatment of multiple high-value indications in fatty liver disease and also Clostridium difficile infection. Our technology has an excellent safety profile. It's generally recognised as safe by the FDA, which allows us expedited review, regulatory review and approval process. We are very well positioned to address unmet medical needs in multiple blockbuster markets that we're playing in. The company itself was registered on the NASDAQ in June 2017, and we have an experienced management team and strong support from key opinion leaders and institutes around the world. Slide 4 shows you our management team. I run the manufacturing and operations group out of our Melbourne headquarters. We have representatives in Israel. Our chief medical officer and clinical director reside in Israel, and we also have a US sales director on the ground in the US. We have attracted prominent scientific advisory board members to our research and development program. Dr. Arun Sanyal from the University of Virginia heads and leads 2 of our phase 2 clinical trials in non-alcoholic steatohepatitis and alcoholic steatohepatitis. Dr. Miriam Voss from the Emory University is our lead PI for our Pediatric non-alcoholic fatty liver disease trial, and we have working collaborations with Dr. Gerhard Rogler focused on colitis from the Zurich University, and we're very fortunate to have Dr. Dina Liris, who is one of the key opinion leaders in Clostridium difficile, and Dr. Dina Liris and her group have really driven our preclinical efforts in this particular project. Our technology platform is based on oral immunoglobulins, and key to this is we focus on specific pathogens, and we try to understand how these pathogens work, how they colonize the gut, and how they cause damage in the gut. And basically we focus on the main key virulence factors, the colonization factor antigens, the flagella, the way the bacteria moves, and also the toxins they produce. And we make proprietary vaccines to these specific antigens and hyperimmunize dairy cows with the vaccines to generate specific polyclonal antibodies, which we harvest from the colostrum, which is the first milk. The colostrum is then purified, and the antibodies generated form the basis of our active pharmaceutical ingredients for our products. The mechanism of action is simple. We target the pathogens in the gut and prevent colonization and clearance, and we also neutralize the toxins they produce. We have a second mode of action, which is the induction of regulatory T cells. The products are designed to work in the gut, and they stay in the gut. They don't actually translocate into the systemic blood supply. One thing we have noted with the technology, we do get a broad anti-inflammatory effect systemically, even though the products work and stay in the gut. So we see an increase in anti-inflammatory cytokines and also a decrease in pro-inflammatory cytokines. But the induction of regulatory T cells also adds to our anti-inflammatory mechanism of action. The platform itself is capable of producing multiple drug candidates to any pathogen we focus on, which offers long-term value creation. We are regulated as a biologic by the FDA, which offers us 12 years exclusivity in the US marketplace for each approved application. And the technology itself is very, very unique, offering protection for future generic biosimilar market erosion. The safety profile is huge, and we are generally recognised as safe by the FDA, which offers accelerated approval process. The slide on slide 7 is a snapshot of our clinical programs and our preclinical programs, which offer multiple near inflection points in terms of investment. We have 3 phase II fatty liver disease studies ongoing. Our non-alcoholic steatohepatitis is the most advanced. We just recently released our interim data, and I'll get to that in a little while. And we hope to see our top-line results for this trial reported at the end of 2017. We also have an alcoholic steatohepatitis trial ongoing. This study is fully funded by the NIH, and top-line results are expected probably early 2019. And we also have a pediatric non-alcoholic fatty liver disease trial, which was initiated at the start of this year. The study is NIH funded and being run by the Emory University, with top-line results expected towards the end of 2018. Behind the clinical programs, we have a very healthy preclinical portfolio, and we're focused on colitis, autism, Shigella and Campylobacter, and also pathogenic ETEC indications, work being done in collaboration with the US Department of Defense. Our second clinical drug candidate, IMM529, has just entered the clinical program, and we have initiated a phase II study, which we hope to see the first patients enrolled early next month. IMM124E is our drug candidate, which is focused on NASH at the moment. Slide number 9 really covers the pathophysiology of the disease, but I'm not going to go through the mechanisms involved. But on the far right side, I will focus on some of the relevant clinical literature that is reported over the last 10 years. And lipopolysaccharide, the endotoxin produced by enterotoxigenic E. coli, has been one of the major factors that really drives inflammation in this particular disease. We also know that Kupffer cells are the key cells in the liver that drive inflammation and fibrosis, and that regulatory T cells play a prominent role in inducing tolerance and anti-inflammatory effects. The drug candidate has been specifically designed to target the endotoxins, the lipopolysaccharide endotoxins, and we see a significant clearance of LPS out of the gut and basically prevent translocation from the gut into the systemic circulation. The product itself also offers an anti-inflammatory response, and it works through the innate and adaptive immune systems, where we see an upregulation of anti-inflammatory cytokines systemically and a decrease in pro-inflammatory cytokines. We also have a significant induction of regulatory T cells, which aid the anti-inflammatory response. The unique competitive profile of our IMM124E really relates to its safety and its unique mechanism of action. And it addresses the multifactorial nature of NASH, and it has significant potential to be a standalone treatment or work in combination with other drugs. We have 3 ongoing fatty liver disease studies in our portfolio. I've mentioned our non-alcoholic steatohepatitis study. Our lead principal investigator is Dr. Arun Sanyal, who is leading this study. We are fully recruited at the moment with 133 patients enrolled with biopsy-proven NASH. The primary endpoint is change in liver fat content confirmed by MRI, and changes in ALT and AST, the liver enzymes. And as I mentioned before, the top-line results are expected at the end of this year. Our other two programs are fully funded by the NIH. The ASH trial is sponsored by the University of Virginia, also headed by Dr. Arun Sanyal. We expect to enrol 66 patients into this study. We're just about over the 50% mark. The primary endpoint is changes in the liver enzymes, ALT and AST, with the top-line results expected in the end of 2018. And our Pediatric non-alcoholic fatty liver disease trial, again NIH funded, is sponsored by the University of the Emirates University. We expect to enrol about 40 patients in this study. Currently, we have enrolled over 10%. I think 5 patients have been randomised into the study to date. The endpoint is ALT changes after 3 months of treatment, and we expect to see the top-line results towards the end of 2018. The mechanism of action we have covered, but in the gut we focus on clearing the endotoxin LPS and decreasing intestinal permeability. We have a systemic effect where we induce tolerance through the innate immune system, and this acts to suppress inflammation. On the serum side, we see a strong metabolic effect with insulin resistance being decreased, circulating LPS being decreased, and an increase in the anti-inflammatory cytokines such as TGF-beta, and a decrease in the pro-inflammatory cytokines such as TNF-alpha. And this all works in the liver to reduce the effect of Kupffer cells, and also we see the reduction of fibrosis and inflammation in the liver. The interim analysis was reported in June, and the goals of the study were really to validate safety and test for futility. The analysis itself was not powered for efficacy due to the sample size being analysed. The design of the interim analysis was initiated when 80 patients in the study reached the 24-week treatment endpoint and had 2 valid MRIs completed. The execution of the interim analysis was performed by an independent committee to keep the company blinded from the results, and the results reported showed an excellent safety profile as expected. The treatment was well tolerated at both doses, and there was no futility recorded. We did see, however, a significant change in ALT and AST after the 24-week treatment period, and this significant reduction was observed over time compared to placebo. We did see a drug-related response, and again, the product was shown to be non-absorbed. It stayed and worked in the gut, and we generated a systemic anti-inflammatory response. This is some of the results reported from the interim analysis, which showed an improved liver function when compared to the 24-week values of serum ALT and AST to baseline. All of the study arms demonstrated a significant reduction in those enzymes. However, the change over time is different across the study arms. And when we looked at the using a logarithmic regression, we noted for both the high dose and the low dose arms, they demonstrated a significant change in liver enzymes compared to placebo over time. So we were very excited to pick up these signals at the early stage of the process. And just to summarise the key milestones for our fatty liver portfolio, our NASH interim results were reported in July this year. We have a number of preclinical studies to support this the clinical program being run with Sanyal Bio Technology and also the Duke University. And we hope to have those preclinical studies reported throughout the course of this year, and early next year we should see us moving into some potential partnering roles to further advance this clinical study. Our pediatric non-alcoholic fatty liver disease study, we expect the top-line results in 2018, as well as our alcoholic steatohepatitis Phase II study with top-line results probably expected towards the start of 2019. I'd like to move to our second clinical asset, which is focused on Clostridium difficile infection. Our IMM 529 is a biologic with a unique triple mechanism of action. We target and neutralise the toxin, the spores and the vegetative cells, and really we are very excited because we see this as a marker designed to basically revolutionise or redefine the standard of care therapy for CDI, which at the moment is antibiotic treatment. The Clostridium difficile is a gram-positive spore-forming anaerobic organism that lives in the gut and actually does all its damage in the gut. And we target the three key virulence components, that is the infectious, highly infectious spores, the vegetative cells which colonize the gut, but also the toxins that they produce which cause all the damage in the gut. We've had a fairly successful preclinical program where we've shown the compound offers at least greater than 70% survival in CDI mice treated with our compound versus less than 7% survival in the control group. The market opportunity for Clostridium difficile infections is forecast to be over 1.5 billion in the year 2024. This current standard of care is vancomycin and metro, and they are basically one of the major issues with the treatments to date is that there is a high incidence of relapse with the current standard of care treatment. Patients that are infected with this disease will see a second reoccurrence in 25% of the population, and once you get a first recurrence, the chance of getting a second recurrence increases to 40% and 30%, 50% for a third reoccurrence. Our product is highly differentiated. It targets the three phases of the disease, as I've mentioned. It targets the highly virulent spores, the vegetative cells which colonise the gut, and also the toxins they produce. And this is really highlighted on slide 22, which shows the mechanism of action where our antibodies work. The preclinical results are summarised in slide 21, and we have looked at a relapse study in mice, and we have shown that treatment with our product, less than 20% of the mice actually have a disease relapse versus nearly 90% of the control groups. In terms of treatment, if we infect mice with the bacteria and then treat, we have an overall 80% survival rate with our particular product compared to less than 7% in the control groups. And in terms of a prevention setting where we can treat mice prior to infection, we have demonstrated an excellent prevention record where 70% of the mice treated with our compound don't get the disease compared to 100% in the control group that all get infected. The program has moved from the preclinical into a clinical setting, and we hope to recruit our first patient in September this year. We are running a randomised double-blinded placebo-controlled study for the treatment of CDI. We are targeting the enrolment of 60 patients, up to 3 weeks of definitive diagnosis with Clostridium difficile infection, and at least 20 of the subjects will be enrolled within the first 72 hours. They will be randomised in a 2-to-1 ratio with active compared to placebo. The patients will have undergone, in conjunction with their standard of care treatment, 28 days of treatment with IMM 529, and they will be followed up for a period of 3 months. The primary objective is obviously to evaluate the safety and tolerability of IMM 529 in this particular cohort of patients, together with standard of care. The secondary objective of the study is to evaluate the effectiveness of our drug candidate together with the standard of care to treat patients with Clostridium difficile infection. The key milestones from this program, the clinical supplies have been manufactured, and we have just opened up a clinical trial site in Israel. We hope to see top-line results coming out of this study towards the end of 2018. Just going to go through some of the corporate and financial overview. We have a very strong patent portfolio with 6 patents in our families, which offer composition and also method to treat claims. We've filed in major geographies, including the US, Europe, Japan, and China, and the patents, the granted patents will end in 2024 and 2013, with the possibility of extending the life of these patents through formulations. There's been quite a significant amount of mergers and acquisition, in particular in the NASH space, which we believe offers potential significant upside to investing in Immuron. And if you look at some of the deals that have been done over the last 4 or 5 years, Big Pharma have definitely had an appetite for NASH assets, and interestingly enough, they seem to be purchasing assets in the preclinical as well as early clinical phase. To give you some idea of our competitors in the NASH and Clostridium difficile space, we'd look at the NASH market, and they really are multi-billion-dollar companies with market caps in excess of $200 million. In the C difficile space, we see similar profiles with companies of market caps of 140 up to $400 million. And compared to our current market capitalisation, as of early August, we were trading at just under about $20 million in terms of our market cap. Our top 10 investors hold about 42% of the stock in the company, with 15% of the stock currently traded on the NASDAQ. As I mentioned at the start, we are fairly unusual for a biotech. We do have a marketed asset, which is a listed medicine and sold in Australia and also Canada. And we generate about $1.5 million worth of revenue this financial year in those markets. Because of the safety profile, we actually do sell the product into the US as a dietary supplement. And we're seeing over the last 3 or 4 months double-digit growth, specifically in the Travelers Medicines clinic in the US. The product itself is labelled and is indicated as a prevention for travellers' diarrhea. And just to summarise again some of the key milestones expected to drive value in the company, we have just initiated our Phase II clinical trial in Clostridium difficile infection, and we hope to see the top-line results of that study reported towards the end of 2018. We have just reported our interim trial results for our NASH study and have ongoing preclinical studies which will be reported throughout the course of this year. The top-line results for the NASH program are expected at the end of 2017, and we also have major reporting milestones into next year with our pediatric non-alcoholic fatty liver disease study and our alcoholic steatohepatitis study, as well as our Clostridium difficile Phase II study. So to summarise some of the investment highlights, we are very well positioned to address some high unmet medical needs in multiple blockbuster markets. We have 2 clinical assets and 4 clinical programs ongoing. We have a robust R&D project pipeline, which will feed into our clinical development program as our clinical assets evolve. We have a validated technology platform with a registered asset generating growing revenue with the potential to expand into new markets. We've listed on the NASDAQ in June and are an experienced management team and have strong support from key opinion leaders and institutes all around the world. So I'd like to thank you for listening to me today, and I look forward to the Q&A session, which is just about to follow this session. Thank you.
Okay, we're now opening the line for our Q&A session with Immuron, ticker symbol IMRN on the NASDAQ. And with us today, we have CEO Dr. Jerry Canales. Welcome.
Good afternoon.
Yeah, very happy to have you here. We'll just jump right into the questions. We only have a few minutes remaining here. Our first question, we've had a couple investors that have asked if you can provide an update on your studies around C. difficile.
Yes, of course. We have actually just completed a fairly extensive preclinical program, and we have got approval to enter a Phase I/II clinical trial. We have just recently opened a clinical site in Israel, and we hope to recruit the first of 60 patients early next month, that is September.
Very good, thank you. And our next question, can you talk a little bit more about your development strategy following your Phase II trial for NASH?
For NASH, the strategy is really to assess the final results. We're hoping to see top-line results by the end of this year, a completed report early next year, and really depending on the results, we have initiated partnering decisions and most probably would move the program forward with a partner. So we're seriously looking at a strategic partnership to advance that program into a Phase IIb study.
Very good, thank you. And our next question, I've had a couple of investors that were curious about Travelan, and if you could talk a little bit about the international expansion and what your marketing strategy is for the product.
Yes, of course. We enjoy a pretty mature market in Australia. We make over $1 million worth of revenue. We have found a unique market segment in the US where we're selling the product through the Travelers medical clinics in the US, and we've actually enjoyed a very, very wealthy double-digit growth over the last few months. So we have seen positive cash flow coming out of the US and hope to build on that and potentially target a $1 million business in the US this financial year.
Very good. Thank you. And we have just a minute or so remaining. I guess in summary, you've covered a lot of great information in the presentation and added some here in the Q&A. But in sum, what's the key takeaway of the Immuron story? Why should investors own the stock today?
I think the key story is we're significantly undervalued for the market segments we're playing in. We have some very, very good company milestones that we'll be reporting on over the 6, 12, 18-month window. So a significant upside in terms of reporting milestones and our share price at present.
Figured quite a lot there at the end there, but very good. That actually takes us to the end of our allotted time. For our audience, this was Immuron. Ticker symbol is, again, IMRN on the NASDAQ. You can learn a lot more about Immuron right here at RedChip.com. And with all the upcoming milestones on some of the trials and other development work that they're doing and the success of Travelan, if you want to stay up to date on those achievements, you can sign up for news alerts right here at RedChip.com, and you'll be one of the first to find out about the developments as they're announced.